I'm Virgile. Let's talk about biofilm. I'm Virgile Guéneau. I started to work with Lallemand as a PhD student between the company and a public academic institute from INRAe, close to Paris, called Michaelis Institute. I'm working in a team specialized in biofilm and spatially organized microbial communities. First, it's a way of life. Biofilm are spatially organized microbial communities with emerging properties in comparison to the free-floating form called planktonic form. It is essential to study them because they are everywhere and they colonize all the biotope on Earth. Microorganisms inside biofilm are embedded in a self-produced matrix that give a three-dimensional structuration of the population. This three-dimensional structuration of population makes the appearance of gradients, making the bacteria that live deep inside the biofilm more tolerant to environmental fluctuations such as the application of biocides or antibiotics. Biofilm can be negative. Most of host chronic infection in animal, plant or human can be associated with pathogenic biofilm. However, they can also be positive and used by humans to transfer food, as during the manufacturing of cheese, for example, or used as biopreservative agent in plant or mushroom. It is described in literature that biofilm can be a reservoir of undesirable bacteria on livestock building surfaces. With the objective of eliminating this biofilm from these livestock building surfaces, cleaning and disinfection procedures are made. However, due to the three-dimensional organization of the biofilm, undesirable bacteria can persist to such procedure and infect animals of the next batches. The classical disinfection technique can leave some sort of empty spaces, allowing fast-growing bacteria to colonize the environment. This is particularly the case of intestinal bacteria that can harbor of pathogens, but it can be also the case of beneficial bacteria from a positive biofilm product. The concept of positive biofilm on farm building surfaces is to apply safe, beneficial bacteria before animal entrance and after cleaning and disinfection procedures. This complex will quickly form a positive biofilm on farm building surfaces before pathogens. These bacteria have been selected to have an advantage against pathogens by many mechanisms involved, such as nutritional and spatial competition. Based on this concept, Lallemand has developed Biofilm Pro, a positive biofilm product to help maintain a safe animal production. Most of techniques used to study environmental biofilm on farm surfaces disrupt the biofilm. But we know that the three-dimensional organization of the biofilm is very important to take in consideration because it's involved in their tolerance. To solve this problem, we have developed a specific methodology, innovative methodology, based on coupon sampling to study the three-dimensional organization of biofilm, the number of bacteria inside it, and the diversity of the population over time. The first objective is to increase the knowledge and awareness about positive biofilm concept and the environmental biofilm on farm building surfaces. The second one is to better understand the mode of action of positive biofilm and create new innovative in vitro model. This model will allow us to select new bacterial candidates and combine them rationally to create the most effective positive biofilm on farm building surfaces. So if you want to learn more, let's visit our website.